The first three process of transport of materials across the cell membrane is passive transport. It's called passive transport. While bulk transport is referred as active transport. Take note. Passive transport is a transport of materials across the cell membrane without use of energy. No energy is spent. Bulk transport, which we call the active transport, is at the expenses of energy. Energy is used to move the material across the cell membrane. So it's very important to understand what happens in diffusion, osmosis, and transport. I know you should be familiar with diffusion. We still do that under the cell and its environment and other topics in biology. Movement of material from region of higher concentration, movement of molecules from region of higher concentration, either water or gas, region of higher concentration to lower concentration, osmosis, movement of water molecules from a weaker solution or dilute solution to a stronger solution through semi-permeable membrane. Transporter system is still diffusion of molecules, but it requires an aid that will help the molecules to diffuse. Remember, gases like oxygen can undergo diffusion across the cell membranes and some other gases as well. So water molecules moves solute potentials of the cells moves across the cell membranes by osmosis. Ions in solutions are transported by osmosis. However, there are certain ions that are transported by the aid of transporter. A transporter is a protein that helps to carry materials across the cell membrane. Example, ion transport channel, sodium potassium pumps. These proteins are called carriers. They carry materials, protein carrier, across the cell membranes. These carriers are known as the transporters. So the diffusion is facilitated, is helped by the transporter. Example, the ion channel. So is aided by a protein that helps to carry ions across the cell membranes. In bulk transport of materials, most materials that cannot move across the cell membranes by cannot be carried by the transporters. Examples, a lot of microorganisms and large molecules which cannot be transported by proteins. They cannot bind to the proteins that transport these materials across the cell membranes. Those larger molecules are transported by the aid of vessels. The vessels are formed by the membranes. The membrane forms a vessel that engulfs the material into the cells. That is endocytosis. Example, it can engulf microorganisms by the process we call phagocytosis. Phagocytosis. So under endocytosis, there are two types. So the endocytosis can be phagocytosis or pinocytosis. To phagocytose means the phagocytic vessels. The vessels, phagocytes, will engulf bacteria or other materials into the cells. Who engulfs this? The vessels developed by the cell membranes will engulf these materials into the cells. If the vessels are developed inside the cells and engulf material out of the cells, we call it exocytosis. Exocytosis. This process requires energy So it is called active transport. In pinocytosis is when the materials, solid materials, we refer them as pinocytosis. So these two processes uses vessels of the membranes, the plasma membranes or the cell membranes, to take materials, digest materials into the cells or out of the cells. They are known as bulk transport because they can carry a lot of materials, large molecules of materials out of the cells or into the cells. They cannot move into the cells, out of the cells on their own because the cell membranes are selectively permeable. So vessels are developed to enable the movement of these molecules, particles, or microorganisms into and out of the cells. But in transporter, there's no vessels. It is a protein that serves as a means of transport, a carrier. It binds the molecules and takes them, mostly ions, into the cells or out of the cells. Those method of transport across the cell membranes is called a transporter or facilitated diffusion. It is a passive transport. And we still have diffusion and osmosis, which I believe you know. So these are the transport mechanisms across the cell membrane. Remember, diffusion and osmosis and transporters are passive transport, while bulk transport is an active transport. That is endocytosis and exocytosis. In transporter, a good example is transport of ions across the cell membrane. Sodium potassium pumps across the membranes of the cells is the example of transporters. Ion channel transporters. These are different. There are different transporters of ions across the cell membranes. So, having known this, now you can be able to draw. You can be able to draw the cell of animal cell. If this is an animal cell, you will know that the nucleus is going to be placed at the center of the cell. And we have the nucleolus and we have the chromatins, right? You can label here the nuclear pores, the nuclear membrane, the chromatins, the nucleolus, the chromatin. All of them is collectively called the nucleus of the cell. The nucleus of the cell. You can decide to draw your mitochondria here. This is the cytoplasm of the cell. If it's a mitochondria, make sure you draw a structure that looks like mitochondria. This is a mitochondrial structure I gave you earlier, is it not? So you call it mitochondria. You can decide to draw your Golgi vessels first. You can decide to call this the Golgi vessels or Golgi apparatus or Golgi body. Under it, you should have just a smaller cell organelles you should call lysosome, which is an offshoot of the Golgi apparatus. Where are we going to take our endoplasmic reticulum? Let's draw it here. Remember the structure I gave you earlier? Something of this nature. Call this place the rough and the other side is the smooth. So this is the endoplasmic reticulum. endoplasmic reticulum. You can draw another structure here and small subunits. What do you call it? With small subunits? Ribosome is called ribosome. Remember, that's what I taught you. It's called ribosome. This is the cell membrane. So outside is the cell membrane. Is the cell membrane. This becomes the basic structure of the animal cell. If I decide to not label it and label them IV, and so on and so forth, you'll be able to identify each of these cell organelles and their functions. They may ask you the function of the part labeled IV is what? You wouldn't know the function if you don't know what the cell organelle is. So that's very important to understand the structures of each of the cell organelle. I can in the same way draw my plant cell. If I draw my plant cell, I will ensure that I put the next one, which is the cell wall. This is the cell wall. The cell wall. I can call this place plasma dermata. This inside is the cell membrane. Plant cell has a large vacuole. I'm not about the vacuole. Vacuole is responsible for storage of water in plants. It contains water. If animals should have a vacuole, it should be small vacuole. If you want to put a vacuole, you say presence of small vacuoles. Here is large vacuole. The nucleus is going to be by the side. You draw your nucleus structure. You draw your mitochondria. You draw your circular part again, which is the chloroplast. Make it double membranes so that we know it's a chloroplast. And put the thylakoid membranes. You draw your lysosome, your ribosome. Draw your lysosomes. You take your Golgi vessels or Golgi apparatus. You draw your endoplasmic reticulum. Your endoplasmic reticulum. So this gives you the structure of the plant. This is the large vacuole. The large vacuole. This is the nucleus. Remember, your labeling should be in one side, just like here. You label in the one side and not label in one side because of want of space. So take note of that. That gives us the structure of the cells. In plants, this structure is made up of this protoplasm plus the cell wall. While here, the entire cells are known as the protoplasm. So this gives us the basic structure of the cells of plants and animals.